Hello everyone, welcome to our last lecture on basic cell structure and function. This lecture will cover non-membranous organelles. So we'll talk about ribosomes, cytoskeleton, and centrioles and centrosomes. So first, ribosomes. Um, so ribosomes, their basic function is protein synthesis. They perform very specifically the final step in the production of proteins. Okay? They are the smallest organelle. You can only see these with an electron microscope, never with like a light microscope or anything else. Okay? And there are two types of ribosomes, what are called bound ribosomes and free ribosomes. So bound ribosomes are ribosomes we discussed earlier when we talked about rough endoplasmic reticulum. They are attached to the outer surface of the rough ER. They produce membrane proteins and secretory proteins, and they feed them into that rough ER. There are also free ribosomes. So you'll see some like orange ribosomes over here that are not associated with that rough ER at all. So these are the free ribosomes. They're not bound to rough ER. These ribosomes are usually in small clusters in the cytosol, and they produce what are called cytoplasmic proteins, proteins that are going to be in the cytoplasm. Um, these ribosomes are only formed when there's an RNA strand for them to produce protein from. So they're not always, if you see them, that usually means that they're actually working. Next membranous organelles is the cytoskeleton. So basic function for the cytoskeleton is to provide structural support, produce cell shape, and participate in cell movements. So there are three types of cytoskeletal elements or cytoskeletal structures. The first are what are called microfilaments. These are made of thin filaments of actin proteins that interact with a thicker myosin filaments, or made of what are called myosin proteins. So microfilaments are made of actin and myosin proteins. And the actin is the thin filament, the myosin is the thick filament. So what you're seeing here is just the actin. You're not seeing the myosin at all, actually. They participate in cellular movement and also in cell division. These microfilaments we'll talk about specifically when we get to our chapter on muscle tissue, because these are the cytoskeletal structures that make muscle contraction possible. The next Sorry about that. The next type of cytoskeletal structures are called intermediate filaments. These are tough, insoluble protein fibers. They have high tensile strength, meaning that they resist pulling forces from their ends, and they're really important in cell-to-cell attachments. So I focus on specific proteins for microfilaments, and I'll also do that for microtubules, but I'm not going to focus on specific proteins for intermediate filaments. So don't worry about a name of a protein. Okay? Next are microtubules. These are the largest cytoskeletal elements. They are stiff but bendable. They are hollow tubes. So notice that this is a big structure. The tube is hollow. Made of these spherical proteins called tubulin proteins. Microtubules are important in determining cell shape. They're used in the movement of vesicles. Remember I told you vesicles can transport things around. That's one of the things that happens on microtubules. And they're also really important in cell division, like mitosis and meiosis. Last are the centrosome centrioles. So centrosomes are spherical regions in the cell near the nucleus that contain a cloud of protein called the centrosomal matrix or centrosome matrix, with an inner portion of a pair of perpendicular centrioles. So notice that this centriole is perpendicular to this one. And they're always like that inside a centrosome. Okay? Now, if you look at a centriole is made of 9 short segments. So notice there's 9 parts. And each of those 9 parts has 3 short sections of microtubules. So centrioles always have this. You can see this with an electron microscope. Whenever you look at a centriole, they'll always have this arrangement, this orientation. So it's always 9 parts with 3 short microtubules. It's never, 8 or, with 4 or anything. It's always like this. Now, the centrosome structures produce and break down the microtubules that are used in cell division in particular. So these are really important in cell division. And it illustrates kind of the importance, they help to illustrate the importance of this lecture as a whole. Technically these are necessary for cell division, and if you see cells, and you will see some cells that don't have centrioles, then that means that those cells can't divide by mitosis. That means that they can't repair themselves, they can't reproduce themselves, because they lack the organelles to be able to do so. All right, so let's answer a couple questions here. So which of the following is made of actin and myosin proteins? Actin and myosin. Right, so remember that's B, microfilaments, right? Microtubules is tubulin. Intermediate filaments, we didn't talk about it. Centrioles, that would still be made of tubulin, because remember, centrioles are made of microtubules.